Chapter 13, Meiosis and Sexual Life Cycles. In the last chapter, you were introduced to cell division, particularly the phases of mitosis and the importance of a properly regulated cell cycle. We learned that mitosis is used by somatic cells to yield completely identical daughter cells for development after fertilization, growth, and repair. In this chapter, we will discuss the second type of cell division, meiosis, and its role in sexual life cycles. In this chapter, you will evaluate asexual and sexual reproduction as they relate to genetic variation. In particular, you will describe and map the roles of mitosis and meiosis in life cycles of plants and animals, describe and relate the relevance of sexual reproduction with recombination and clonal asexual reproduction in the context of evolution, and you will be able to relate meiotic non-disjunction in genetic karyotypic variation. Before we learn the specifics of meiotic cell division, we need a thorough overview on topics relevant to the sexual life cycle. In the last chapter, we learned that living organisms are distinguished by their ability to reproduce their own kind. All cells, including but not limited to animal cells, plant cells, bacterial cells, and fungi, are all living organisms that are able to reproduce. If you recall from the very first chapter of your textbook, we covered the 5 unifying themes of biology: organization, information, energy and matter, interactions, and evolution. We learned that the unifying theme of information relates to the life processes that involve the expression and transmission of genetic information. This was our first introduction to DNA and genes. In this chapter, we begin to dive deeper into this unifying theme to fully understand the role of genetic information in the continuation of life. To begin, we must examine how chromosomes pass from parents to offspring in sexually reproducing organisms. This underlying mechanism eluded biologists until the advancement of genetics in the 20th century. Genetics is the scientific study of heredity and inherited variation. Heredity is the transmission of traits from one generation to the next. I'm sure you already know that you inherit everything from your hair and eye color to height, muscle structures, and the number of fingers you have from your parents. Yet you are not an exact copy of either of them. And if you have siblings, you do not look exactly alike, and this is due to variation. Variation is demonstrated by the differences in appearance that offspring show from parents and siblings. In this unit, you'll learn about genetics at multiple levels, from organisms to cells to molecules. So, what exactly are you inheriting when we talk about heredity? The answer is genes. Genes are the units of heredity and are made up of segments of DNA. I want to repeat that. Genes are segments of DNA. Revisiting Chapter 1 again, we learned that the DNA gene sequence provides the blueprint for making a protein. We learned in Chapter 5 that proteins include a diversity of structures and result in a wide range of functions within the cell. Genes are passed to the next generation via reproductive cells called gametes, which are sperm and egg. In the last chapter, we learned that gametes are our sex cells, the sperm and the egg. We also learned that a typical human somatic, non-reproductive cell contains 46 chromosomes, 23 from mom and 23 from dad. Chromosomes are how the DNA is packaged within the cell. Always remember, the cell is a highly organized piece of biological machinery which has been fine-tuned over the last 3.5 billion years. As such, each gene has a specific location called a locus on a certain chromosome. We'll touch more on this in the coming chapters on genetics. It's important to note that there are 2 types of reproduction, asexual and sexual. Any instance where 2 parents give rise to offspring that have unique combinations of genes inherited from those 2 parents is called sexual reproduction. Examples of sexual reproduction include certain stages of fungi, plants, and of course, animals like humans. In contrast, asexual reproduction is when a single individual passes genes to its offspring without the fusion of gametes such as egg or sperm. Examples of asexual reproduction include binary fission of bacteria and budding in yeast. A clone is a group of genetically identical individuals from the same parent cell. You often hear this used in cell culture, specifically while growing bacterial colonies. You'll see more of that in microbiology courses. But let's get back to chromosomes. Recall from the last chapter that human somatic cells are any cell other than a gamete. This includes bone, heart, lung, or kidney cells, just to name a few. Somatic cells all contain 46 chromosomes, which is 23 pairs. We say it like this because these cells received a set of 23 from mom and a set of 23 from dad. The 23 from mom and 23 from dad are matched sets, and they have a special name. The 2 chromosomes in each pair are called homologous chromosomes or homologs. This is a very important term. Chromosomes in a homologous pair are the same length and shape and carry genes controlling the same inherited characters. Take a look at the karyotype pictured in the bottom right. A karyotype is an ordered display of the pairs of chromosomes from a cell. Take a moment to confirm that all 23 pairs of chromosomes are pictured for this individual. This is a great way to understand homologs. Each pair of chromosomes is a homologous pair. Let's focus on pair number 1, indicated in the white box. Both chromosomes are the same length and shape, and they also carry genes for the same inherited characters. For example, let's say hypothetically that chromosome number 1 carries the genes for eye color and hair color. Both of these chromosomes carry a gene for eye color and hair color, and they will be in the same place on those chromosomes called the locus, or loci for plural. One of the chromosomes in this pair was inherited from the mother and the other from the father. There are 2 types of chromosomes, sex chromosomes and autosomes. Sex chromosomes are the pair of chromosomes that determine the sex of the individual, called X and Y. Human females have a homologous pair of X chromosomes, so they are XX. Human males have one X and one Y chromosome. Can you tell by the karyotype of this individual whether they are male or female? In the bottom right of the karyotype, we see the sex chromosomes X and Y. This individual is a male. Notice that the X and Y chromosomes are not the same size. The Y chromosome only carries an estimated 100 to 200 genes, while the X chromosome carries around 1,000. The remaining 22 pairs of chromosomes are all called autosomes. Again, each pair of homologous chromosomes includes one chromosome from each parent, and the 46 chromosomes in a human somatic cell are 2 sets of 23, one from the mother and one from the father. Cells that have 2 sets of chromosomes are called diploid or diploid cells and are indicated by the symbol 2N. For humans, our diploid number is 46. So if 46 equals 2N, then what does N equal for humans? We'll get back to that in a moment. Here is a representation of 3 pairs of chromosomes. The red chromosomes are all from the mother, while the blue chromosomes are all from the father. In this cell, the chromosomes have replicated already, so there is actually twice as much DNA in this cell as a normal cell would have. Recall from the last chapter that when a chromosome duplicates, the 2 identical copies remain tethered together by the centromere. The 2 identical chromosomes are called sister chromatids. Look at the longest blue chromosome pictured. This blue chromosome is comprised of 2 sister chromatids held together by the centromere. Now look at the longest red chromosome. This red chromosome is comprised of 2 sister chromatids held together by the centromere. You can see it is the same for all 6 chromosomes pictured. Now here's the part that often trips up students. Which chromosomes in this picture are homologs? Remember, homologous chromosome pairs are chromosomes that consist of one from the mom and one from the dad. The longest blue and longest red are one homologous pair. The medium-sized blue and medium-sized red are another homologous pair, and the smallest blue and smallest red are the final homologous pair. The diploid number for this cell is 2n equals 6. Therefore, n equals 3. 3 of the chromosomes are from mom, and 3 of the chromosomes are from dad. Don't confuse sister chromatids, which are just identical copies for the sole purpose of creating a new cell by cell division, with homologous chromosomes, which are the pair of chromosomes from your mother and father that are the same size and shape and carry genes for the same characters. Now you see that a diploid cell has 2 sets of chromosomes, one set from the mother and one set from the father, and is represented by 2n. The diploid number is different for each species. Our diploid number is 46, so what is our n, or haploid number? For humans, n equals 23. When a cell contains just one single set of chromosomes n, it is said to be haploid. The only haploid cells we have are our sex cells, or gametes, the sperm and the egg. We'll see why this is important in a moment. Each set of 23 consists of 22 autosomes and just one sex chromosome. For an egg or ovum produced by a woman which has a genotype of XX, the only sex chromosome it can carry is the X chromosome. Sperm cells, on the other hand, may carry either an X or a Y. Why is this? This is because men carry the genotype XY. This is why it is said that men choose the sex. All eggs give an X, and depending on whether the sperm that fertilizes the egg is carrying an X or a Y, will determine the sex of that individual. This union of the gametes, the sperm and the egg, is called fertilization. Once an egg has been fertilized, this cell is called a zygote and is the very first cell in the creation of a human life. Take a look at the human life cycle shown. All multicellular organisms begin as just a diploid single cell. That zygote is able to produce somatic cells by mitosis and develops into an adult. At sexual maturity, the gonads, which are our reproductive glands, the ovaries and the testes, produce haploid gamete cells, the sperm or the egg, via meiosis. Gametes are the only types of human cells produced by meiosis rather than mitosis. It's important to remember that meiosis results in haploid cells with just one set of chromosomes in each gamete, and this is denoted as N. When the 2 haploid cells, the sperm and the egg, meet during fertilization, the chromosome number is restored to 2N in the formation of the diploid zygote. You can see that fertilization and meiosis alternate in sexual life cycles to maintain chromosome number. So how does meiosis maintain chromosome number within the life cycle? We now have the foundation to begin talking about how this is accomplished. Like mitosis, meiosis is preceded by the replication of chromosomes. Here we see a pair of homologous chromosomes. The red is the maternal chromosome and the blue is the paternal chromosome. During interphase of meiosis, the homologs duplicate and the sister chromatids are shown held together. Remember, sister chromatids are genetically identical and joined at the centromere, while homologous chromosomes are genetically diverse, but are the same shape, size, and carry genes for the same characters. Replication happens during S phase of interphase. Before a cell proceeds through meiosis, all chromosomes are duplicated like this to produce a diploid cell with duplicated chromosomes. Meiosis takes place in two sets of cell division called Meiosis I and Meiosis II. In Meiosis I, the homologous chromosomes are the ones that separate. We see that the maternal sister chromatids went to the right, while the paternal sister chromatids went to the left. In Meiosis II, the sister chromatids are the ones that separate. The two divisions result in 4 daughter cells rather than 2 daughter cells, which results from mitosis. Each daughter cell has only half as many chromosomes as the parent cell. The result is 4 haploid cells with unduplicated chromosomes. This was just an overview of Meiosis I and Meiosis II. Now we're going to dive a little deeper into the specific phases and key events that occur. You'll recall from chapter 12 that there are 5 phases in mitosis. They are prophase, prometaphase, metaphase, anaphase, and telophase. Mitosis is then followed by cytokinesis. As we move through the phases of Meiosis I and Meiosis II, I will only be focusing on the aspects that are unique to meiosis and are important for genetic variation. If you need a refresher on the basics, please check out the video I posted on chapter 12, because understanding the phases of mitosis will make learning meiosis much simpler. Meiosis I and Meiosis II each contain 4 of the same phases as mitosis. Division in Meiosis I occurs in 4 phases: prophase I, metaphase I, anaphase I, and telophase I, and then is followed by cytokinesis. Notice how all 4 phases are followed by the Roman numeral I. This is to denote that we are discussing events in Meiosis I rather than in Meiosis II. Prophase I of Meiosis I. Prophase I is one of the most important phases of meiosis. It typically occupies more than 90% of the time required for meiosis. The pairs of sister chromatids begin to condense, and something very significant called the tetrad forms. The word tetrad means a group of 4, and in this case, a tetrad is a group of 4 chromatids. The 4 chromatids are the 2 pairs of homologous chromosomes binding together. This binding is called synapsis and is a precise pair-up of the homologous chromosomes whereby they align gene by gene. I want to stress how important this formation of the tetrad is. There is no other time in cell life or division where homologs, the mom and dad version of chromosomes, are able to interact with each other. The reason why this is so important is because this allows for a special process called crossing over to occur. Crossing over is an exchange of DNA between non-sister chromatids. Remember, sister chromatids are completely identical when we begin, so exchange of DNA between them would result in no difference at all. However, the homologous mom and dad version of chromosomes contain different variations of characters. For example, your mother may carry the brown eye gene for the eye color locus, while your father may carry the blue eye gene for that eye color locus. At the synapsis, these genes have aligned, and the chromosomes can cut and paste the DNA segments to switch the eye color traits. This place where the cut and paste occurs is called a chiasma, or chiasmata for the plural. Chiasma comes from the Greek word meaning to cross, and chiasmata are X-shaped regions where crossing over occurs. There can be as many as 20 chiasmata along a pair of homologous chromosomes in a tetrad. In this image, we see 1 pair of homologous chromosomes which has been duplicated. The pair of homologs have synapsed and 2 chiasmata have formed. These are the 2 X-shaped regions shown. Crossing over occurs at the chiasmata. Crossing over produces recombinant chromosomes, which simply means that the inherited parental DNA has recombined. We see after crossing over that 2 homologous chromatids each have pieces of the other. these are now recombinant chromosomes. Two of the sister chromatids, the ones on the outside of the tetrad, did not exhibit crossing over. Therefore, these are said to be parental chromosomes because they contain the DNA inherited originally from the parent. Anaphase I of meiosis I separates the homologs, and anaphase II of meiosis I separates the sister chromatids. What do you notice about the 4 daughter cells? Are they the same? How are they different? We'll return to this later. Metaphase I of meiosis I. During metaphase I, the tetrads line up at the metaphase plate with one chromosome facing each pole. Just as in metaphase of mitosis, the microtubules emanate from the centrioles of the centrosomes and attach to the kinetochores of the chromosomes, one from each pole. During metaphase of meiosis, another special event called independent assortment of chromosomes occurs, and this has to do with how the tetrads line up at the metaphase plate. During independent assortment, the homologous pairs of chromosomes orient randomly at the metaphase plate. In other words, each tetrad has one side of chromatids from mom and the other from dad. When the tetrads align, they randomly arrange. Take a look at possibility 1 and possibility 2. We have 2 tetrads in each cell. In possibility 1, the tetrads randomly assorted so that all paternal chromatids will separate to the left and all maternal chromatids will separate to the right. In possibility 2, one of the tetrads is flipped so that some maternal and paternal DNA will end up in both cells. Looking at the daughter cells in possibility 1 and comparing them to the daughter cells in possibility 2, we see that just that one change in how the tetrads randomly assorted changed the genetic material in the daughter cells. It created more variation. Now imagine this in a cell that has 23 tetrads lined up along the metaphase plate. The number of combinations is astounding. You can calculate it with the formula number of combinations equals 2n, where n is the haploid number. For humans, our haploid number is 23. Therefore, there are 2 to the 23 possibilities, which is 8 million possible combinations of chromosomes possible due to independent assortment. This variation comes solely from the ability of the chromosomes to assort independently of one another along the metaphase plate during metaphase I of meiosis I. Anaphase I of meiosis I. In anaphase I, pairs of homologous chromosomes separate. Sister chromatids remain attached at the centromere and move as one unit toward a pole. Telophase I of meiosis I. In the beginning of telophase I, each half of the cell has a haploid set of chromosomes, with each chromosome still consisting of 2 sister chromatids. Simultaneously, cytokinesis is separating the cytoplasm to form 2 haploid daughter cells. In animal cells, a cleavage furrow forms. In plant cells, a cell plate forms. No more replications occur because the chromosomes have already replicated prior to meiosis I. At the end of meiosis I, 2 non-identical haploid daughter cells are formed. Each cell has 23 chromosomes, 46 chromatids. Make sure you understand this distinction between chromosomes and chromatids. To figure out the number of chromosomes in a cell, we actually count the centromeres. Because the sister chromatids are still attached at this point via a centromere, the 46 chromatids count as 23 chromosomes. These cells are therefore haploid as they only contain one set of chromosomes. Now the cells move into meiosis II, which also occurs in 4 phases: prophase II, metaphase II, anaphase II, and telophase II with cytokinesis. We learned how unique and important Meiosis I was, with important events like formation of the tetrads, crossing over, and independent assortment occurring. Meiosis II is much more similar to mitosis. Prophase II of meiosis II. Prophase II is just like prophase during mitosis. The chromosomes present in the cells are sister chromatids attached to each other by the centromere. The centrioles of the centrosomes form the spindle apparatus as those centrosomes move to opposite ends of the cell. The microtubule spindles attach to the kinetochores of the chromosomes and begin to move them toward the metaphase plate. Notice there are no tetrads and no crossing over here. That was special to prophase I. I also want to bring your attention to one important observation here. In mitosis, the sister chromatids are completely identical copies of each other. Is that true of the sister chromatids in prophase II? No. These sister chromatids are no longer identical because of the crossing over events from prophase I. Metaphase II of meiosis II. In metaphase II, the sister chromatids are arranged at the metaphase plate. Because of crossing over in meiosis I, the 2 sister chromatids of each chromosome are no longer genetically identical. Therefore, the way they orient can create more genetic variation. The kinetochores of sister chromatids are attached to microtubules from both opposing poles. It is crucial that both sister chromatids attach to spindle from opposite sides of the cell, otherwise the 2 sister chromatids will not be pulled apart during anaphase. Anaphase II of meiosis II. In anaphase II, the sister chromatids separate and move as newly individual chromosomes toward opposite poles. Movement is due to the kinetochore. Telophase II of meiosis II. In telophase II, the chromosomes arrive at opposite poles of the cell and the nuclei begin to form. The chromosomes will also start decondensing. Simultaneously, cytokinesis separates the cytoplasm. At the end of meiosis II, there are 4 daughter cells, each with a haploid set of non-identical chromosomes. Every single one of these cells is unique and genetically distinct from the others and from the parent cell. In learning mitosis and meiosis, it's important to be able to compare and contrast the 2 types of cell division. Not only does this help deepen your understanding, but exam questions are often presented in a way that tests your ability to distinguish between the 2 processes. Mitosis and meiosis are similar in many ways, yet differ in a few essential points. A comparison chart is a great tool to add to your notes, like the one you see here. During your study process, ensure that you understand this chart and would be able to recall information from it easily to answer various types of question prompts. Let's review this comparison between mitosis and meiosis. First, mitosis is the cell division process used by somatic cells to produce 2 identical daughter cells, while meiosis is the cell division process used to produce 4 non-identical daughter gamete cells. For both mitosis and meiosis, DNA replication occurs at S phase of interphase. Mitosis consists of 1 division of the DNA, while meiosis consists of 2 distinct divisions of the DNA. In this way, mitosis conserves the number of chromosome sets, while meiosis reduces the number of chromosome sets from 2, which is a diploid, to 1, which is a haploid. In addition, Meiosis has 3 unique events, and all 3 occur during Meiosis I. The first unique event is synapsis and crossing over, which occurs during prophase I. During this event, the homologous chromosomes physically connect to form a tetrad and exchange genetic information at chiasmata to create recombinant chromosomes. The second unique event occurs at the metaphase plate during metaphase I. The tetrads consisting of the pair of homologous chromosomes line up and orient randomly in an event called independent assortment. And the third unique event occurs at anaphase I, where it is the homologous chromosomes instead of the sister chromatids that separate. This is in contrast to both mitosis and meiosis II, in which the sister chromatids are the ones that separate. All of the special events of meiosis have one purpose, and that is to create genetic variation among individuals. Genetic variation in sexual life cycles contributes to evolution. You probably already know that mutations, which are changes in the organism's DNA, are the original course of genetic diversity. Mutations due to chemicals or radiation create different versions of genes called alleles. We'll talk more about alleles in the next chapters. An example of different alleles are the blue eye gene for eye color and the brown eye gene for eye color. Reshuffling of such alleles during sexual reproduction produces genetic variation. 3 things aid in the reshuffling of alleles, and they are independent assortment of chromosomes, crossing over, and random fertilization. We've already discussed the first 2, independent assortment of chromosomes and crossing over. Let's discuss the last one, random fertilization. Fertilization is, of course, referring to when the male gamete and the female gamete fuse to create the zygote. It's called random fertilization because there's no way of knowing which sperm will fertilize which egg. Therefore, any sperm cell from the male can fuse with any egg cell from the female, which increases genetic variation. Remember, for a human, there are 2 to the n possible combinations of chromosomes due to independent assortment for an individual. That means 8.4 million possibilities. The fusion of 2 gametes, each with 8.4 million possible chromosome combinations, produces a zygote with any of about 70 trillion diploid combinations. That number is just from independent assortment. Crossing over adds even more variation. Each resulting zygote therefore has a unique genetic identity. You've now learned mitosis and meiosis. Try to answer the questions that follow in the lecture PowerPoint. And in the next chapter, we will learn about Gregor Mendel and the origin of the gene idea.